Okay, good afternoon, everyone, and thanks for being here. I know it's after lunch, so I'll try and speak loud so you can hear me and keep awake. So firstly, I'd just like to thank CHPC for inviting me to have a short discussion on the research that I do, and as well as the CSIR for hosting this event. So as mentioned, my name is Dr. Pritika Ramharack, and I am a senior scientist at the South African Medical Research Council. And today I'll be discussing a bit of the tools rather the research techniques of cheminformatics and how I apply it within the African traditional medicine context, or rather South African phytochemicals. So firstly, just a bit about the South African Medical Research Council and those that have trypanophobia. I'm sorry about the circles. Just a bit about the institution. The SAMRC was established in 1969 to conduct as well as fund health research, innovation, development, as well as knowledge transfer within South Africa and surroundings. So the main research focus of the SAMRC falls within the 10 most prominent disease states that leads to mortality, comorbidities, as well as risk factors in disabilities. So the research focus deals with a lot with laboratory techniques, as well as public health and clinical research as well. So the SAMRC actually has numerous research units and platforms that are based all over the country in different provinces. I'm employed by the Biomedical Research and Innovation Platform, and we are situated at the Medicina campus in Cape Town. So within our unit, we have about 25 employees. And as you can see, of those 25 employees, we have a huge research niche. All of this is done by our 25 employees, and the different research niches include hypertension, cardiovascular disease. There's been a lot of research done on rooibos, especially in regard to diabetes. And we've also done a lot of preeclampsia work, as well as obesity and dietetics. A few up and coming research niches at the moment is gut health. And recently we've been involved a lot with wastewater surveillance of SARS-CoV-2. So that happened when the pandemic started. We also have a lot of research specializations, or rather techniques that are established within the unit, including LCMS, DNA methylation techniques, as well as advanced tissue culture. So the two that I'm particularly involved in is the Cheminformatics, and I'm heading up the African traditional medicine within the unit as well. Okay, so other than our employees, we also have partnered with multiple universities, and we host postgraduate students by firstly supervising, funding, and training them within our facility. So this is a bit about my team. One of them at the back is here today. The rest of them are all joining online. So we have Ms. Karen DeBase, and she's doing her PhD in Mycobacterium tuberculosis therapy. Ms. Nolo Qabiso Mangwane, she's doing SARS-CoV-2 work and the different variants that are currently circulating. We have Ms. Ane Lerma, she's actually within clinical pharmacology and doing work there. And Doctors Kolofelo Malemela and Nonhle Sangweni, who are looking at natural products and phytochemicals and how they affect the pharmacokinetics within a system, especially looking at cytochrome P450 enzymes. We have my master's students, which is Ms. Andre Kutzer. She's looking at anti-cancer therapies and fetal programming. And then our little rose that we have here amongst the thorns is Mr. Kwandile, and he's heading up the ATM work using a three-phased approach. So just to discuss a bit about the African traditional medicine system. So currently, South Africa hosts 11.5% of the global flora that's found, and 80% of the South African population actually use these traditional medicines either alone or in conjunction with chronic medication to treat various ailments. So the adverse effects of these traditional medicines at the moment is that there's no standardized technique or no standardized methodology when looking at research within ATMs. And at the moment, Africa is lacking a pharmacopoeia where these natural plants or these plants have been identified, bioactive compounds have been found, and the mechanism of action is known. So this is not the case for, if you look at the Chinese pharmacopoeia as well as the European pharmacopoeia, it's very well established, but at the moment, this is a problem we're having within Africa itself. So another adverse effect of these ATMs is that it leads to herb-drug interactions. So when taken with chronic medication, it actually affects the metabolism of the drugs, and it could then lead to pharmacokinetic effects. Either the drugs don't work, they become resistant to the chronic medication, or it could lead to toxic effects in the body. So part of my funded research within the SAMRC is to come up with a standardized biological testing pipeline that can be used for African traditional medicine. So I know there's a lot of research groups within South Africa that are doing research on these natural extracts. However, there are different methods and different means of testing the different extracts. So we are trying to find some sort of streamlined process that everyone could use, that we could then generate into a pharmacopoeia. I know the South African Natural Compound Database is doing an excellent job at the moment of putting these compounds or identifying compounds and collating it within a database. But we still need to do more on our end to ensure that we are getting this database and finally a pharmacopoeia ready. So at the moment, I'm looking at, so we've got a few of the extracts through the pipeline, and we're hoping to get eventually to clinical trials. And at the moment, what we're concentrating on is disease cases or metabolic disease cases, looking at diabetes, obesity, wound healing, and cancer. So I know this is a lot, you see a lot of experimental techniques happening there. So where my work fits in would be after extract characterization. So once we have identified these compounds within an extract, they may be bioactive compounds or the most abundant compounds within the extract that we could then take further. And what we do in that instance is go through the Cheminformatics workflow, of which the identified compounds are then put through a pharmacokinetic and toxicological profiling through online tools. So that I'm sure if any of you are within the same domain, you must have heard of Swiss ADME and Protox before. Those are normally the first go-to softwares that we use. We then can identify the biological targets. So if you know that an extract has anti-diabetic effects, for instance, there are softwares that would allow you to then see within the anti-diabetic effects or the pathway of diabetes, which of these biological targets does this drug act on, or does this compound act on at that point. The next phase would be molecular docking. So molecular docking offers a geometric fit. So it takes the compound, and I don't know who, can I just have a quick hand of who's chemistry or biologically inclined? Okay, sorry to those of you that aren't, I'm very sorry. So what you do is we take the compounds and we put it, or we place it where it would act on an enzyme rather. So just the orientation of where it fits. That's just geometrically, there's a space, the compound fits there, and that's that. It doesn't mean that it's going to act on that enzyme. So that's why we go over to molecular dynamic simulations. And this is where CHPC comes in. So what happens in this instance is that we take our drug through docking and we put it into the enzyme, and we take that complex and we simulate an experimental environment. And during that environment, we then allow the protein as well as the compound to become flexible, and there are molecular interactions that occur, and that's when we actually see what is happening when that compound is interacting with the enzyme. After that, we then move back. It's either if the drug, I keep on saying drug, but if the compound works well, great, we can move to synthesis and in vitro testing. If we are seeing there's adverse effects in these compounds, or the interactions we're seeing are not the most optimal, there's an option then to biochemical techniques to then optimize the molecular groups on the compound that may lead to better interactions and have elicit a better effect. So with regard to that, this is where CHPC, just explaining the molecular dynamic simulations. So Currently, that's the number of CPU hours that we've used in 2022 alone. So it's sitting at the million stage at the moment. And that's just a few of us that you saw on the slide previously that are using that much of time. The reason being we deal with enzymes as well as the drugs and obviously looking at an atomistic level. And I think Paulo explained earlier where you look at coarse-grained versus atomistic, and then you get to the quantum. So we're not looking at the quantum, we're not looking at the electrons, but we are at that atomistic stage where every atom within the enzyme and the drug is taken into account during these simulations. So what happens is, so we use CHPC, we access it, and then we use a software called the AMBER software suite. So that is used specifically for biological systems. And from there, we take the drug or the compound and we would separate them initially after docking and then put them together in the simulation. At the correct conformations, we would then add a solvent as you would do at a wet lab work level. We charge it and then we make sure that all the parameters for a standardized experiment or within physiological conditions are met. So that's a temperature, a physiological temperature to ensure that we are meeting all the parameters required. After all of this has been set up, we then allow the simulation to run. Normally, we would deal with at least over 100 nanoseconds of the simulation to get to allow for the entire enzyme to become flexible and for interactions to happen. And after those 100 nanoseconds, we then run our post-analysis, where we would then see of the enzyme what amino acids are fluctuating. And again, I'm sorry to those of you that don't have this background, but we would look at when the compound is acting on the enzyme, what is happening at that point, what interactions are happening, what molecular interactions are being formed, is there hydrogen bonds, hydrophobic energy, and that will give us a feel of whether the drug is or the compound is working or not. Okay, so this is just an example of one of the projects that we're currently working on. So this is my pet project at the moment, and it is characterizing the pharmacophoric features of bufadienolides from Drimia species as anti-cancer therapeutics. So as you can see here, the Drimia species is actually a bulbous plant, which is located in South Africa. It's from the Aspergillus family, and it is found in various locations within South Africa, including Northwest, Free State, Gauteng, and the Northern Cape. So these plants have been found to have multiple beneficial effects, including antimicrobial activity, anti-inflammatory, antioxidant, and anti-cancer properties. So although they have all these, because they have all these therapeutic effects, they are now also found on the red list by SANBI, which is our South African National Biodiversity Institute, and they are now found on the red list. That means they are endangered because of overharvesting at that point. So they're used in a lot of traditional extracts, and therefore they are being overharvested, and we are running out of them very quickly. So it's important in this instance to identify from these plants what therapeutic compounds there are in them, or bioactive compounds, and if we are able to mass synthesize these compounds. So can we produce it synthetically instead of going to now get the plant and extract it and then get eventually to the point of having a therapeutic effect after being through all the toxic effects of the extract itself. Okay, so the way we look at it is, in this instance, there is a family of compounds that are known as bufadienolides, and they're found in the Drimia species specifically. They're also known as cardiac glycosides. And what they do is they act on the pump. So in layman's terms, in cells, there are pumps that allow for sodium into the cell, or sodium out of the cell, sorry, and potassium into the cell. And that leads to an exchange in your pH and harmonious cell activity. So what happens in cancer conditions is this ATPase pump actually becomes overexpressed and leads to the cancerous conditions. So there are these bufadienolides that have been shown to have anti-cancer effects through the ATPase pump. However, the problem with this family of compounds is that at high doses, they are extremely toxic. And that is seen with a lot of well-known compounds such as digitoxin. So the aim of the study was to identify the key molecular groups that are found in the bufadienolides that are required for the inhibition of the sodium-potassium pump. So I think I explained all of this before, so I'm not going to go too in depth. So the first thing we did is we got the compounds, and that was through literature as well as the South African National Compound Database. And then we got the 3D structures of the proteins, and that again is very chemistry driven. It's either we get the 3D structures through X-ray crystallography or NMR, and then we acquired the 3D structures of the compounds. We then moved on to molecular docking, and finally through the simulations. From those simulations, we measured the free binding energy calculations and assessed the binding landscapes of the multiple compounds. So as you can see, we started with 53 compounds. After docking, I filtered down to 14. And finally, after the free binding energy calculations, we ended up with 4 compounds that we generated a structure activity relationship from. So this is, as I said, after we run our simulation, this is the RMSD plots that we get. So your root mean squared deviation through the 150 nanosecond simulation, and it basically shows the stability of the complex over time. So there's a time where the complex will minimize or will get to a point where it's not comfortable. And when it gets to a point where it's become flexible enough and is just vibrating, it will reach convergence. And that convergence is generally over here, I think it's around 60 nanoseconds, and then you'll have a plateau that forms. So that's what you want to see in the graph. Over here, we have an exception of compound 5. So from the Gibbs free binding energy calculations, which may hit the mathematician's notes here, just so that's one of the calculations we use to measure the free binding energy, and 4 of those compounds of the 14 all had favorable binding energies compared to the known drugs, Obein and digitoxin. Okay. So finally, we managed to do, and I know it's really small here, but we managed to put together ligand interaction plots. And if you kind of see, you can see, I always call them the red eyelashes. So the red eyelashes you see on the sides here leads to hydrophobic energy, and that happens within the compound. So hydrophobic energy from the amino acids lead to energy to the compound, and that's what stabilizes the compound within the active site of an enzyme. So based on those interactions, we figured out that this particular molecular group here was required for activity to occur. We found out that there had to be hydrophobic interactions that are happening within the steroid structure of the compound. We also realized that we do need a side chain group that is added to the scaffold molecule, which would lead to then hydrophobic and hydrogen bond interactions. And we need two hydrogen bond donators at carbon 12 and 14. I know to most of you, this is very foreign, so I'm just going through it very fast. So basically, we came up with the structure activity relationship. We just realized we need to remove bulky groups or the side chains that are not necessary, that are not leading to interactions, as they may be leading to the toxic effects we are seeing in a clinical system. So at the moment, we're just going to look to screen the compounds and see what's out there, if there are compounds that are synthesizable, or do we need to synthesize or optimize compounds. We're then going to go through the drug discovery pipeline and hopefully get something out that we could work with as an anti-cancer therapeutic. Okay, so that's my acknowledgements. Thank you, everybody, for inviting me here. I appreciate it. And my funders as well. And then finally, I just have to do this, but Krishna has been a huge part of our work from Prof. Soliman's days in 2016 when I just first started. So I know he's moved on. I wouldn't say greener pastures, but he's moved on. But I just want to thank him. So wherever if he's online or wherever. So thanks to Krishna. Thank you.
Thank you very much. Thank you very much. So are there any questions? Yes.
Okay, thank you for the nice presentation. My question is, are there chances that a compound cannot give positive docking results, but give a very impressive in vitro and in vivo?
Yes, that is very possible, and it's very dependent on what you're using then as a positive and negative control. So as you would with in vitro, you should always have a positive and negative control in all in silico work in this regard. So then you would match up where the compounds of interest that you're looking at, where they fit in within your positive control as well as your negative control in that regard. So it's also very possible in some instances, if docking does not work. I know, for example, and I'm going to use this as a big example, ivermectin, for instance. Ivermectin is such a large molecule, it does not fit into any of the cytochromes through metabolism. However, we do know that it does act on a cytochrome. And that docking won't tell you much about it, because remember, you're just taking a rigid enzyme, you're taking the rigid compound, and you're just placing it. So it's sometimes, if you're getting adverse or negative results from docking, it's always good to take it to simulations nonetheless and assess thereafter.
We have time for a couple of other questions.
Thank you much, Doctor, for a good presentation. I would just like to ask, what are the criteria are you going to use to choose an enzyme? Because I believe that if you just taking the enzyme and putting it, it acts as an enzyme and a substrate, they had to bond in some way. So which criteria are you using to choose an enzyme that will work best on your research?
Okay, so there's two ways I can answer that. So firstly, the identifying an enzyme, firstly, there's specific software that works in a way where you would add your compound, and it's based on your SMILE, so based on the molecular structure of your compound. You would add that onto the website, and it's programmed in such a way that it would identify compounds that are similar, that are found in chemical libraries. And based on that similar scaffold, it will then identify targets that are known, that have been shown in vitro or through in vivo work, that act on those similar compounds. And it's a matter of then predicting how, or the probability rather. So how probable is this compound to act on that enzyme? And then secondly, when you're choosing your enzymes by criteria, you would use protein data banks. So that's one of the big ones I use. There's also UniProt and various others. But it's also based, X-ray crystallography is always, for me personally, is the best option. You get your best resolutions on that. And that's just forming the 3D crystal structure you require.
Any other questions? Yes, quick, last.
It's not much of a question, but I just wanted to ask, is there a reason why in all of this you did not screen, so you did not do any virtual screening?
Yeah, so the whole point of the study was to identify compounds within these plants that are endangered, firstly. So at this point, and it was on my last slide, we will then look at the screening. So based on the pharmacophore or the model or the scaffold that I generated, we'll screen for compounds that are out there. There may be a compound that's already produced out there that could have an anti-cancer effect that it hasn't been looked at in that manner. So there is the screening aspect that is yet to come. Yeah.
Okay, if there are no other questions, let's thank Pritika again.
Thank you so much. I'm so sorry to the non-chemists.